Moving around large chromosomal segments will give a big advantage to synthetic biologists. Welcome to Microbial Minutes, ASM's update on what's hot in the microbial sciences, the 2 chromosomes are better than 1 edition. I'm Julie Wolf, Science Communication Specialist here at ASM, and today we're going to be talking about a paper from Science that demonstrates a new CRISPR application that enables genomic sections. Now, the ability to specifically move around pieces of DNA has enabled all sorts of iterative edits of different types of genes or genetic loci. Think about like the ability to produce opioids or cannabinoids within microorganisms. CRISPR has allowed scientists to both disable certain genes through making a double-stranded break, and that's often repaired by non-homologous end joining, which will often disable the gene, as shown on the schematic there. Or if a new piece of DNA is added, additional gene functions can be added as well. But this is often something that takes a whole number of genes for a large metabolic pathway, such as making those cannabinoids that I mentioned. And the technology so far has allowed really the movement of single genes or genetic loci. The ability to move large pieces of DNA around will be important for a field such as synthetic biology that is increasing the size of the chemically synthesized genomes that it is able to make, as well as moving around those large metabolic pathways. And that's one of the reasons that this group decided to tackle this problem. The idea that they are coming up with, moving around pieces of chromosomal DNA, is one found in nature, as bacteria can often undergo chromosomal transversions, chromosome splitting, chromosome fusion. And what this group wanted to ask is, can we engineer these sequences to rearrange in ways that we want them to? And so they did this using the workhorse of molecular biology, E. coli, and broke the single circular chromosome of E. coli into 2 chromosomes by adding first a fission back, a bacterial artificial chromosome. And this back, which I'm showing you in the schematic here, contains a number of genetic determinants that help it to be a useful tool. It has things such as a sucrose sensitivity, chloramphenicol resistance double selection cassette, streptomycin sensitivity cassette, a Lux operon, which allows the cells to be luminescent, and of course, an origin of replication that allows this piece of DNA to be passed on to progeny cells. During fission, chromosomal fission, Cas9 is used to break the DNA backbone where the black arrows are indicating within the image here. And that's going to split the E. coli genome into 2 fragments and the back into 4 pieces. The back comes with homology markers to the genome where it's been split, and this allows lambda red recombination, which is a method of homologous recombination, to join the fragments and generate the 2 new chromosomes. In this image, you can see that the specific recombination of these genotypes is confirmed within the phenotypes that is shown in panel B, where you can see that pre-split and in 2 iterations of the chromosomal rearrangements, they are able to see a difference in things such as the Lux operon activity, where luminescence is dimmed after the chromosomes have been arranged, where sucrose sensitivity has increased, and the other patterns which have changed also. They use a similar process of this chromosomal rearrangement to show that they can chop up the E. coli genome in a variety of different ways, all dependent on the homologous fragments that they include in the back. And this allows them to bring in a synthetic biology angle by then rearranging the genome of a recoded E. coli, which has had several codons reprogrammed. Finally, they combine the genomes of different E. coli strains and demonstrate that they can generate entirely new lineages using this programmatic rearrangement. On the next slide, we'll see that the work was highlighted in Science itself, where Anne Meyer, a synthetic biologist who was not related with this research, is quoted as saying, "The technique will enable synthetic biologists to take on grand challenges, such as writing information to DNA and storing it in a bacterial chromosome, or creating new hybrid bacterial species that can carry out novel metabolic reactions for chemistry and materials production." Imagine the ease by which complex biosynthetic pathways can be moved between strains and possibly between species. This is a very exciting step for synthetic biology. To hear more about synthetic biology, please check out our Microbial Minutes about the recoded E. coli genome, which we covered a few months ago. We'll include a link down below. Be sure to hit subscribe for future updates on synthetic biology. By turning on notifications, you will never miss a video. You've just heard about an application for CRISPR technologies that allows cutting and pasting of entire chromosomal fragments. I'd like to thank you for listening and thank Ray Ortega for production. I'm Julie Wolfe, and I'll be with you next time on Microbial Minutes.